It's my pleasure to introduce to you Ilaria Panzeri, Dr. Ilaria Panzeri. She's a research assistant in the Department of Epigenetics at the Van Andel Institute in Grand Rapids. She received all of her education, at least up to her PhD, overseas, which is lovely. She has her BS in biotechnology, an MS in biology, and a PhD in translational and molecular medicine. And all of those were from the University of Milano Bicocca. Okay, in Milan, Italy. And then she did postdoctoral work at the Max Planck Institute of Immunology and Epigenetics in Germany, and then came to the United States. She also has been, came to the United States to the Van Andel specifically, and is in their Department of Epigenetics there as a research scientist. So anyway, she also has been awarded numerous prizes and honors, including the Human Frontier Science Program Fellowship, which I think is probably pretty hard to get, pretty competitive. So we're very happy to have her here. On a human note, she has a few extra little tidbits. I do know she's married and has a little boy. So maybe we'll learn more here. And I really appreciate everybody coming today and taking part in this. And so enjoy what we get to learn.
So hopefully, yes, my mic is on. Good morning, everyone. Thank you all for being here today. I would like also to thank the organizers, the tech support, and everyone for having me. And I just want to go over a bit what I will present today. So I will give a bit of background about who I am, what is epigenetics. I try to keep everything simple. I know we have different expertise in the room, but please feel free to interrupt me if anything is not clear, if I'm going too slow, too fast. So I'm flexible. So next step would be what is cancer, then epigenetic and cancer, putting them two together. Number 5 would be a bit what I do, new avenues in cancer epigenetics. And number 6 will be a bit about the epigenetic therapy that now we have to target cancer. So as Jane presented, I'm from Italy, and particularly from a small village here. I don't know if you know the area. Let me see if I can move the cursor here. So it's a very small village near Lake Como, on the shores of Lake Como. And this is how it looks like. It's nice. I mean, I'm biased, but it's a nice place. So if you have plans to go to visit the area, to visit Italy, I will be happy to share tips, maybe during the break. And Jane shared already a bit of my background. I did most of my undergraduate and graduate studies in Milan. And then I moved to the Max Planck Institute in Freiburg, so very close, where I got a Human Frontier Science Fellowship program that I brought with me at the Van Andel when the lab moved. So I did a bit of my postdoc in Germany and a bit here in the US. And here I also won a pilot funding to generate some more data that I will need for my next step of the career. So while I was in the United States, actually about 2 years ago, I had a boy, a beautiful boy. His name is Leo. We tried desperately to find a name that didn't sound like mafia, together with the last. So hopefully it's fine. And this is how it looked like back then. He was supposed to be born May 4th. So we were all, hyped about, I don't know if you saw Star Wars movies, so May the 4th be with you. But no, he decided to be born May 5th. So anyway, yeah, exactly. And a bit about our hobbies as a family. So we really like to hike. And of course, Michigan is the perfect place to hike. And we've been hiking with Leo since he was very little. So now he enjoys it, doing everything by himself. He's 2 years old. He's like, Mom, I can do it. And of course, being Italian stereotype, we also love to cook. And these are some traditional dishes that we make, and this is my husband making homemade pasta. So if you want some recipes of something, I'm also happy to share. So now back to science. So again, I kept it very simple. Feel free to interrupt, to say, oh no, it's too slow, whatever. Okay, so I want to start from very basic biology. So when I studied, there was this central dogma of biology that stated that um, eukaryotic cells had DNA material, so the genetic material contained inside the nucleus. And this genetic material is constantly replicated every time the cells divides. So this ensures replication of the genetic material in each and every cell. And also, there are basic units in the DNA sequence that code for mRNA or messenger RNA that is transcribed based on this very defined and limited set of DNA sequence. And then this gets translated into the protein. So DNA and mRNA are usually found in the nucleus, then mRNA gets exported in the cytoplasm, so inside the cells but outside the nucleus, where it gets translated into protein, then the protein folds and exerts its function. This was the central dogma of biology. In reality, now we know it's not this simple, like most of the things in biology, and it looks more like this, a mess. So mRNA in general can also be retrotranscribed, so it can become genetic material, and this is it's done by viruses, for example, that invade our genome. mRNA can replicate itself, can act by itself through non-coding RNA, so it doesn't necessarily is not necessarily translated into a protein. It can function by itself. And actually, the machinery that generates the protein is composed by RNAs, so it's super complicated. So basically, we learned that, to question everything that we used to know in the biology. And another thing to think about is I mentioned that DNA is packed in the nucleus, right? But reality is DNA is a molecule that is 2 meters long, right? Here I just picked a guy from Hope College. I don't know if some of you know him. Apparently he's a star in basketball and he's about 2 meters tall, right? You can imagine that has his opponents think, how do I shrink him? Similarly, the cell needs to shrink 2 meters long DNA in 20 microns nucleus. So it's quite a shrinking, right? So a cell faced this dilemma during development. How do I shrink the DNA? How do I even fit this thing inside me? And the second dilemma that they face is how can DNA, if we have a long string of instruction that is the same and it's identical in every single cell, how can it be that we get many different cell types in the body, right? A liver cell has the exact same genetic material as a brain cell. So how does it know to be a liver cell or a brain cell? So reality is DNA is not present in a linear naked form. So it doesn't look like this. It looks more like this in most of the times in the cell life. So very compacted. And the most compacted form is the chromosome that is present when the cell divides, to be sure, to replicate and share with the daughter cell the proper genetic material. And to be so condensed, DNA is wrapped around proteins that are called histones, that are this sort of beads. And indeed, the DNA is called beads on a string. So it's similar to a necklace, right? That it's folded over and over again, like when you travel and they all get clumped up in the pores, right? So, but how is compaction modulated? So it actually changes. It's not that all DNA every time is compacted the same way. Actually, a cell most of the times looks like this, where you have very dark and very condensed DNA molecules that are called heterochromatin, and very decondensed and open molecules that are called euchromatin. And in reality, these two phases, it's what dictates which genes are read at which moment. So how a cell knows to be a specific cell type. Because heterochromatin is highly compacted, so nothing can enter, right? Genes that are in these compacted states cannot be read. So for example, liver gene cannot be read by brain cells if they are in this compacted form. So that brain cells doesn't develop into a liver cell, just a brain cell. Conversely, the brain cell will have open chromatin, so accessible chromatin, in brain-specific genes, so that these are activated in brain cells. and the cells doesn't activate liver genes, for example. So here comes the specificity of the cell type. And as I mentioned before, genes that are open are accessible to RNA polymerase, that is the enzyme that produces messenger RNA, then it's transcribed into protein, and then the brain cell knows to be a brain cell. So you can imagine DNA as a list of words where every word is a gene. If you read all of the words, they don't absolutely make sense, right? And that's not what it happens. What it happens is that you have highlights here and there in the DNA molecule in the text that tell you hepatocytes, so liver cell, needs to produce carbohydrates, lipids, and protein to detoxify metabolism, activate immune cells to maintain liver homeostasis. So this is how the DNA book is read by a liver cell, by highlighting what's relevant for a liver cell and not highlighting what's relevant, for example, for a brain cell in the liver context. Because the liver cell doesn't need to be a brain cell, right? And the same is true across multiple cell types. So, but how does, I made this example, of course, epigenetic is not a true highlighter in a text. So how does it look like? It looks more like this. So here I'm giving you just two examples, but again, we have many other layers of regulation. So I mentioned that DNA is compacted and wrapped around proteins. These proteins are called histones, and they are modified in some residues biochemically so that can be read as open or close. Go and compact, silence this gene, or open, that makes this gene accessible. Similarly, in the DNA molecule itself, in the actual text, we can highlight certain words through this DNA methylation. And whenever this DNA methylation happens, these words is not read. We can mark it in black, whatever, no read here. Whenever DNA methylation is removed, it's actually a highlighted yellow. Now you cell can read this text. So epigenetics is an active process. It continuously changes. And there are writers that add these highlights. There are erasers that remove these highlights. And there are readers that actually read the highlight when it's there. So it's a continuous dynamic between these three processes. Yes, and I will get back. Yeah, the question was if it changes during a lifetime. It's a great question, and we'll get back to this later. Okay, so first example, DNA methylation. So this is an unmethylated DNA. This is normally read, and transcription happens. And so this gene is active. Whenever the DNA is methylated, then it's covered. You can imagine like it's covered, and so it cannot be read. And how methylation happens for you that you that are into chemistry is by the addition of a methyl group in the cytosine residue in the DNA, actively by specific enzymes. So this is how DNA methylation looks like. So as I mentioned, this matches what I told you before. So this is how the cell look like. The unmethylated residue are the open form where transcription can happen, produces mRNA, produces peptide, and the protein folds. Methylated regions are instead darker, so closer, more compacted, and transcription doesn't happen. So as I mentioned, epigenetics is a highly dynamic process, and it looks more like this. So there are enzymes that are DNA methyltransferases that impose the methylation, so they add the methyl group to the cytosine. And then there are enzymes that remove actively the methylation. So it's a constant circle. It constantly happens. But DNA methylation can be also stable. So if we need to silence stably a gene, for example, a brain cell never needs to become a brain cell. So these will constantly be there in liver cells, right? In liver genes. Whereas during development, as has been asked, these processes are more dynamic, and I will again get back to it later. So the second example that I give you are histone modifications. So again, they are like highlights that are added into the protein that tell them if they need to be more compacted or less compacted, thus exposing more or less the genes that are covered by them. And in reality, this is more or less how they look like. So these beads are actually composed by smaller beads, and each and every one of these beads can be modified biochemically by specific enzymes that are listed here, and each and every one of these modifications has a meaning. So I'm giving you just a couple of examples. So there is acetylation, so the addition of acetyl groups to the histone that is usually an active mark. This means that whenever histones are acetylated, they cannot stay close together. They tend to open and then expose the gene that is in between them for transcription, production of protein, and expression of the gene itself. Methylation, once again, is instead most of the times a repressive mark. This tells histones to become more and more compacted, more close to get together, one to the other, and therefore the gene that is in between them cannot be read, and therefore it's silenced. So once again, open chromatin, so acetylation of the histones looks more like this, open, accessible. Methylation of histones looks more like this, darker, more compacted, less accessible. I said most of the times, because in reality, this is how histones look like and how methylation or modification happens. There are many more. So there is methylation, there's acetylation, there's ubiquitinylation, and we are discovering more and more again. And it's in reality the combination of all of these modifications that are read at a given time by the cells to understand what to do with this protein, if to compact genes or not. So it's a very complex and dynamic process. And some of these modifications are mutually exclusive. So we know that if one happens, another one in the same residue cannot happen, right? So why does this matter? So I want to give you a key example that is sex determination. So in humans, sex is determined by sex chromosomes, that in females are two X chromosomes, and in males, it's an X and a Y chromosome. In reality, what we know is that it's not that both X's are expressed, because you can imagine that here we have the X gene that expresses the gene, the Y gene the Y chromosome, I'm sorry, that expresses the gene. Here we have two X's, so does it mean we have double the amount of genes from the X chromosome? In reality, no, because we know that actually gene dosage is really important. There are diseases that emerge due to the presence of multiple copies of a chromosome. So we know that this needs to be carefully kept in check. And females don't have double the expression of X in this case, because this may cause problems. So in reality, what happens is the inactivation of one of the two X's during the early development. So at the very beginning, we do have the expression of both. Then randomly, what happens is that we have an expression of one gene from one of the two called exist. And these genes induces the compaction of the more or less the entire chromosome where it is expressed. So one of the chromosome gets silenced and the other one gets expressed. So this ensures proper balance so that female can have, just as it's supposed to be, one of the two, only two chromosomes expressed. And this is how it looks like. Again, sorry, it's got, again, open and closed chromatin. So in green are both X chromosomes activated in the cell life. This is what happens when one of the two starts to express the exist RNA, so it starts to begin the process to become inactivated. And this is how it looks like when it's inactive. It's very interesting because I don't have time to mention this now, but normally in a cell, all the genes or most of the genes that are inactivated are stored in the periphery, and genes that are expressed are stored in the middle. So you can see that the more, during the X inactivation, what happens is that it really goes towards the periphery where it gets silenced. Whereas the other one, you don't see it because it's cropped, but stays in the middle and it remains expressed. Oh, I think I have it here. Here it's more clear. You see, this one is here, and this one is the periphery. So this one is expressed, this was not. And I mentioned that the inactivation is random, and this is actually relevant for the phenotype. So if you've ever seen cats like this, these are called calico cats, and they have these patchy colors on the fur, and these derive from random X inactivation. So in cats, you have this gene that codes for the color of the fur attached to the X chromosome. So depending on which X chromosome gets inactivated, you will have some of the fur that is yellow, some of the fur that is black, and some of the fur that is white, depending on this random process. So it really has real consequences, even for cats. So as someone asked before, epigenetics is changing during development. And this is how the process usually looks like. So starting from germ cells, so sperm or oocytes, what happens is that in reality, epigenetic marks are completely erased, particularly DNA methylation during early development. And then they are reestablished in the genome deriving from the sperm or the oocytes at different time points. Here is the puberty, fertilization, and early implantation, the zygotes, so the merge of the maternal and paternal um, genomes. And then over and over the development, you see that now we are losing again the mark up until the blastocyst, that is before the specification of the different organs in the embryo. And then the marks are reestablished once again. So this ensures that at every generation, we have a refresher of the epigenetic mark, but not for all genes. There are some genes that are stably marked during development, and they are inherited from father and mother. These are called imprinted genes. And as you can see here, their epigenetic status is maintained, and they are really important because they regulate differentiation and body development, and they are also associated with diseases. So whenever this process is altered, we may get some disease. So epigenetics is also a good way to respond to the environment. You can imagine that if I have a DNA, so if I have a textbook that really needs to remain there and be untouched, because if words change, then it will become a mess. It's better to have changes that happen on top of this. That's what epigenetic means, right? Changes that happen on top of DNA without altering the DNA sequence. And this is exactly how epigenetic works. It responds to the environment, where environment can also be development, for example, can be anything. So here is a prototypical example for honeybees. So honeybees, they have the exact same identical genome. Despite this, they develop in different castes that have different functions across the colony and different appearance. So queen is responsible for reproduction of the colony. Workers are responsible to work, as the name implies. And they are very different, you see, in terms of shape, body size, but also relationships within the colony and behavior. And the reason why they develop like this, again, the genetic material is absolutely identical between these animals. It's epigenetics because it depends on what they eat during early development. Depending on the diet, epigenetics, so how we read the genes changes. And so bees develop in different forms with different function and different behavior. A similar example is the agouti mouse. So these mice have different, again, coat color that ranges from yellow to brown. And this depends on the methylation, again, DNA methylation epigenetic status of the agouti gene that encodes for coat color. So whenever this gene is unmethylated, is expressed, and then the mouse turns yellow. Whenever this gene is methylated, these genes is not expressed, and therefore it turns brown. And this is also associated with the health status of these animals, where yellow animals are more prone to diabetes and obesity, whereas darker animals are overall healthy. And this depends on the diet they are eating, because again, you can think that methyl groups needs to come from somewhere, right? And diet can be enriched or depleted of methyl groups or dietary derived methyl groups. And therefore, if you eat a lot of methyls, then you may end up like this. If you don't eat enough, then you may end up like this. And so again, epigenetics is a way to respond to the environment and express differently the same exact genetic background depending on the environment itself. This also means that for human, during development, there are some very sensitive windows that we need to be careful and aware about, in which any sort of environmental exposure can have consequences, even if it doesn't target the DNA, but it targets just epigenetics. And that's, for example, the reason why for pregnant women now folate is recommended as a supplement to make sure that all these methyl groups happen in the right place at the right time. Folate-deprived diet can be linked to disease later on for the fetus and for the newborn. So this is really important, and it's an area that's been studied throughout the year, but we still lack some very key information about these processes, especially with regards to the disease. One example of this is the so-called Dutch famine. So there was a period in the Netherlands where unfortunately kids and mothers were exposed to famine, so they weren't receiving enough nutrients. And epidemiological studies had this weird observation that kids that were born or from pregnant from mothers that were pregnant in that period and were born later actually were developing as obese or having metabolic disease. How can this be the case if they were undernourished when they were born or right after that? Well, in reality, what we discovered is that undernutrition during early development or in the mother causes transition to obesity, hyperglycemia, and other metabolic diseases later in life. And this is due to epigenetic changes happening in early life. So it's really important to be aware of these processes to properly, feed the mother and understand how to track disease later on in life. So I want to take a step back and compare a little bit genetic and epigenetic changes. So as I mentioned, changes can happen in the DNA sequence as well. We call these mutations or variants. And of course, those are stable, right? Can be passed from one cell to the other through the replication that I mentioned at the very beginning. And so they are hardly subject to change. So it's really hard for the cell to fix them. And these can create long-term processes, long-term problems that are passed through the generations. So whenever the body needs to change something to adapt to the environment, that's not a very reliable and safe solution. And so the easiest way that the cell has to adapt to the environment is instead through epigenetic changes that doesn't target the DNA sequence, just how genes are read in the genome. So it's highly dynamic. It changes in response to the environment, including development, can be readily reversible most of the times. There are some epigenetic modifications that are more stable than others, but cells have a way to change them. It affects multiple genes at a time, so it can act all over the genome, as I mentioned before. For liver cells, it tells liver cells what to do, what to produce, when to develop, so multiple genes at a time. It's heritable, because as I showed you before, epigenetic changes in the mother can affect the baby, but it's also modifiable. So we can also, from our perspective, from the perspective of a therapy, it's much easier to act on the epigenome than to act on genetic mutations. So I have a bit of summary on this part, just to make sure we are all on the same page, and please ask questions if you have. So we said the DNA is not present as a linear sequence in the nucleus, but rather it's packed, and this ensures that it can fit inside the nucleus itself. Apart from this, it also ensures that it can be read differently despite being identical in all cell types. And these mechanisms that pack and read differently the cells are called epigenetics. And the definition comes from Greek, and it means on top, epi, of the DNA sequencing itself. So thus allowing the read to modify how genes are read without changing the DNA sequence. Some of the key epigenetic mechanisms, not all of them, we have many more, are DNA methylation and epigenetic and histone modifications. Both act as opening and closing the DNA so that some genes are accessible where some others are not. Epigenetic is modifiable and changes during development. So it's a constant ongoing process that responds to the environment, while genetic changes are instead permanent most of the time. Epigenetic is impacted by the environment. It's actually a way for the cell to respond to it and for the body to respond to it. And some examples are the diet, as I mentioned, folate for pregnant mothers, but also pollutants. So microplastics are one key examples that we are reading over and over again now. Asbestos is another one that can act directly on the epigenome and so on and so forth. And epigenetic is inherited and have long-term consequences. And this is super important because we need to understand that our choices in life can impact the next generation, and we need to be aware of that. Even though when I was pregnant, I just craved pasta. So, okay. Do we have any question? Yeah.
At one point, you mentioned folic, and I think folic was also folic acid.
Yeah.
And if it's missing, an infant can be born with some severe problems. Honduras has this problem. And a man that was guiding me around Honduras said if Coca-Cola would put folic acid in their bottles, just a bit, it would prevent this. Would that be possibly a concept?
That's an interesting idea. Yes and no. So the problem of, in general, those supplements is that we need to be careful how much we take them as well, because too much can be too much, right? And we don't need that much amount of folate throughout our entire life, just in those windows of sensitivity. That's why I feel it's better and safer to, give them as supplements rather than in everyday.
And that's in pre-birth.
Yes,
The mother needs to have
specifically,
that
yes,
supplement.
Yes.
Well, they're drinking
Of
a lot
course,
of
there.
Coke.
Yeah. Yeah. And also Coke comes with other things like
Right,
sugar
right.
or
But that's
so-
because the water is not good there in these small
yeah.
villages.
Yeah,
That's why.
That's it.
And I have one other
That's interesting.
quick
Thank you.
question. You said obesity is caused from low birth weight, possibly,
Yeah, among other
and
things.
can be changed. Is that why some obese people opt to have surgery to reduce their stomach, or is it?
So that process is called bariatric surgery, and it's recommended for highly severe obesity, not for the general obese population. And that's a way to treat the disease when it's so established that other methods are likely to fail. Because also you have hormonal derangement. So it's really hard, oftentimes, unfortunately, obese people hear things like, oh, you should just stop eating, or you should just exercise more. In reality, that's not true. That's not how obesity works. It's a real derangement of hormones in the body so that you don't have control over these things, oftentimes. But that's kind of disconnected to the let's say, intergenerational consequences, because also most of the time obese people have problems with infertility. So it's a complicated scenario, but I wouldn't connect that to the Dutch famine. That was an example, and it's not necessarily true that if you're born underweight, then you will have obesity. There are association studies that are epidemiological data, and we know that obesity is a very complex and heterogeneous disease with many different causes. So it really depends on the situation. That was just to give an example of how much diet and pre-birth can influence long-term outcomes, but it's not a one-on-one relationship. It doesn't necessarily. Thanks.
Inheritance of changes from one cell to the daughter cells would involve instructing enzymes to how to acetylate or methylate various things. I'm wondering how is this transmitted?
Yeah, so great question. I didn't go too much into details, but that's exactly what happens. So there are readers, enzymes that read this epigenetic modification and basically copy this into the new DNA that is produced in the next cell. We're still studying which type of epigenetic modification are actively passed from one cell to the other, because as I showed you, there are so many, and we don't even know all the enzymes that read and copy all of these. But that's exactly what happens. So for example, for DNA methylation, that's what happens. Some regions of the DNA methylation are read and copied in the newly produced DNA molecule. But we know that for some histone marks, that's not so immediate. So we miss a bit how then the newly produced DNA knows where to put the mark. We still don't know. It's still something that an area that it's under active study, but it's interesting. Thank you. Okay.
Yeah. We'll get there.
Yeah.
So first, a comment about supplementation and then a
Yes.
question about histones.
Yes.
The idea of supplementation has been around for a long time. And there's been a lot of debate in the US about supplementing snack foods because people eat a lot of snack foods. The FDA has been resistant to do this with good reason. And you alluded to it, too much of a good thing can be a bad thing. So we should never see supplementation of snacks, whether it's a beverage or solid food, for those reasons. It would be too much administration into the diet in some cases. The second thing about histones, you've talked about them repeatedly. Can you tell us more about what they are? Do they differ from cell type to cell type? And a little bit more about their function.
Yeah, sure. So let me go back so it's clear. Thanks for the note on the supplements. I didn't know that the FDA was discussing about. Okay, so histones are actually proteins. So you can imagine them as proteins that really bind to one another. You can see this. They are called octamer because there are 8 different proteins that complex one to the other, like this. They are called in different ways. So they're called H2A, H2B, H3, and H4. That's just the name of the protein and how they combine. And there's one that's so-called linker histone that is called H1 that binds all of these 8 together and merges them together. And as I mentioned, all of these proteins, you can imagine them as, I had it here, as a list of, sorry, we're going back and forth. Here, as a list of amino acids, so a list of molecules that are attached one to the other, and they derive from a specific, again, messenger RNA. So the process of their production is similar to any other protein, and they fall together to form this octamer that then binds, that then binds the DNA, where is it? Here. That then binds the DNA. So these proteins form, they go together, they form this octamer, so 8 proteins together plus the linker. And then our DNA is wrapped around it and compacted around it, depending on how these different molecules of the protein are modified. So these residues that compose the protein, especially in the so-called tailed that are exposed, a bit more exposed compared to the center, can be modified with the chemical modifications that I mentioned before, so methylation, acetylation, and so on and so forth. And these are imposed and read and erased by other proteins and basically modulate how much the DNA can bind because of affinity and interaction, chemical interaction. And they are different across different cell types, and that's what modulates the cell identity. And this doesn't only mean that they are differently marked or modified in different cell types, they're also differentially compacted between cell types. And we also know, to make story even more complicated, that most of these proteins can exist in multiple variants that are different across cell types. And also the frequency, you can see, as I mentioned, like a beads on a string, right? The frequency of the beads on the string is also different between cell types and also during a cell life or an individual life. There can be more or less beads, more or less close one to the other. more or less open relative to the other. So you can really think that like every cell has its own necklace composed of these beads on a string that looks different and exerts different function, basically. Thanks.
I'm one of the old-fashioned learners of biology back in the days.
Okay.
So I appreciated this whole discussion and the way you introduced it. It's just great. So cancer cells are cells that are no longer as differentiated as they should be. And one of the first things in their de-differentiation is they lose the ability to say, hey, I've got enough neighbors, I'm going to stop dividing.
Yep.
Instead, they lose that ability and they say, well, I'm going to keep dividing despite the fact that I'm bumping into lots of other cells around here. Is that a product of this methylation or demethylation and histone action as well?
Yeah, you're scooping my next part.
That's
Yes,
gonna be
the
part
answer is
two.
yes,
Yeah.
yes. It's exactly how one of the ways through which cancer doesn't adapt anymore to the environment, it just overgrowth and forgets about everything, right? Epigenetic is one of the key mechanisms, but I will go back to this later. Thank you.
My ears peaked up when you mentioned pollutants, since I'm a toxic tort attorney, retired. And epigenetics is just now entering our field where IR, International Agency on Research into Cancer, is coming out with some research on what pollutants actually are causing it in thousands of people who
Yeah.
have thousands of claims.
Mhm.
Yesterday I attended a thing on PFAS, and I've been wondering, PFAS, that big pollutant, could it cause an epigenetic cascade, and is that why it's causing cancer in all these different organs?
Yeah, it can be. So this is an area of active research. It's relatively recent. And as you can imagine, because of the flexibility of the epigenetic system, it's really difficult to rule out the true connection, particularly in human life. We're exposed to so many things that to draw an actual connection is very hard. But yes, that's one example through which pollutants can act. They alter, it's known that some of them alter really how the enzyme can access the different proteins, the modifications that they can impose. They act in so many ways. They change the metabolites present in the cells, so the availability of the methyl groups, the acetyl groups, to be able to, that the proteins can attach to these residues. But it's an area of research that is still under study. I think I'm not an expert on PFAS, but I believe there are some examples of it in the epigenetic field. Yeah, unfortunately.
Oh, I guess we have one more question.
It's sort of a two-part. So all those variables, that's just astounding. So do you assign that all to a computer sheet, or how do you track all that? I mean, on a daily basis?
Yeah, that's a great question.
Or
I-
one individual basis?
Yeah, that's a
One
great
person.
question. Yeah. So I didn't put too much detail about, technically what we do, but for sure to disentangle each and every single of these components, you need to have systems in which you can keep constant one of the things at least, right? So for example, being able to compare individuals that have the same genetic background, such as monozygotic twins, and look at how the epigenetic changes during their life. It's a way to keep, you have the same genetic material, or bees, like the example that I showed you before. You have a similar genetic material, so you can kind of focus on something else. Of course, human is a particularly difficult example because we all live in different environments, exposed to different things. For animal models, that can range from insects, worms, mice, and so on, and plants, and so on and so forth, you can better control all the variables. So this means that you can raise these models in very tightly environmental condition, changes one thing and see what happens, right? You need to have a way to simplify the model, as you said. And that's also one of the reasons why we don't know everything about all of these things, because it's, we need to kind of target them one by one in order to understand the biochemical processes behind them. But we know they are not read by one. Then we need to add another one and see how they interact with one another, and so on and so forth. So it's a long and complicated process, but
One last question. So how do they, this is another section you were talking about. You were talking about, some of that in terms of the epigenetics, sometimes you make the choices or something, especially food or something
Yeah.
like that. And then they become hardened and they turn into heritable.
Yes.
So are you tracking the things that cause them to be j- heritable?
Yes.
And you'll
So,
get into that maybe later in your,
yeah.
okay, but I'm.
So for example, there's a study that's ongoing in the lab where we're trying to understand how different diets impact that the mother eats impact subsequent generations. So what we are doing is we are feeding, in this case, animal models with different diets and look at the pups and how they develop. And there's a way to trace the metabolites, so to mark the metabolites and see where they go in the body, so that we are really able to trace what they do and how stable the changes are, because we know that some are more stable than others, and which genes they impact, and so on and so forth.
I'm just going to make a comment that you can
Yeah.
elaborate on, I think. So, in my mind, I always thought of the Van Andel as being cancer oriented, but they actually have recruited a whole team of experts in epigenetics. And they actually on their website talk about trying to accomplish an epigenetic moonshot,
Yeah.
which would include not just cancer, but also metabolic disorders and things like that. So this is a very complicated field, and we probably can get more speakers back now
Yeah.
that you're laying the groundwork for us, which is very interesting, obviously, to this group. Would this be a good time to take a break, do you
Yes,
think?
I
You
think
know?
there was another question here. I don't know.
Oh.
Okay. Okay.
You can.
Uh,
should I-
I'm available over the break. and-
Okay, all
Okay.
right, thank you.
you.
All right. So we'll reconvene in like, I don't know, 10 to 15 minutes, please. That work? Okay, well, I think we're going to reconvene part 2 of our lecture now that we have some basic science under our belts. I want to reassure everybody there will not be a quiz, so you're all safe. And Ilaria has graciously consented to us having a copy of her slides. And so I think what you're going to get is a copy of her slides and the video recording of the class, because some people are having trouble with their hearing loops and can't hear adequately to get, and we don't do closed caption in here. They have that at home, but not here. So anyway, stay tuned. You'll be getting more information you can review if you want. And now our second part is cancer and the impact of epigenetics on cancer. So take it away.
Thank you, Jen. I haven't put it in the slide, but feel free. I will share maybe my email. So if you have any questions, just feel free to shoot an email and I will be happy to answer later. Okay, so let's jump into cancer. Again, a bit of background. What is cancer? So I will start by the classical view of cancer. So cancer starts in the classical view whenever a genetic mutation or variant hits a cell, and particularly when it hits genes that are associated with an increased risk for cancer. Then hyperplasia, so overgrowth and messed up growth start, so hyperplasia and dysplasia. The cancer becomes an in situ cancer, so it's limited in space and it's not invasive at this point. And later on, it can progress into invasive cancer and enter the circulation and spread through metastasis. This is the classical view of cancer. Cancer is usually classified in broader terms as liquid or solid cancer, but then it's classified by type, so cell of origin, and by location. So we can have, for example, carcinoma, if they are targeting epithelial tissues, lymphomas, if they are targeting the lymph nodes and the lymphatic system, sarcomas, if they're targeting the bones and the soft tissues, myeloma, for example, if they are targeting the plasma cell, and leukemia, if they are targeting blood-forming tissues. And there are many more subtypes of cancer, and basically each and every single type of this broader type of cancer can be then classified depending on the tissue that it's impacting. So you can have a breast carcinoma, or you can have a lung carcinoma, and so on and so forth. So this process of oncogenesis, here I'm showing you, and the example that I'm showing you is for solid tumors, but liquid tumors work similarly, not exactly in identical ways. But this process is overall accompanied by the acquisition of hallmarks of cancers that are skills that the cancer cell needs to acquire in order to sustain oncogenesis. And the classic view of the hallmarks of cancer is this one. So it's a list of capabilities that the cell need to acquire. So for example, we know that cancer cells tend to overgrowth. Like someone mentioned before, they just go on and continue growing no matter what the environment is. They fight death. So of course, there are mechanisms inside the cells that whenever something goes awry, tends to stop the cell from growing, tends to kill the cell. But cancer cells have ways to avoid this. Cancer cells also modify the host in order to get nutrients. So oftentimes in tumors, we see increased angiogenesis, so development of new vessels that bring nutrients to the tumor so that it can grow. Cancer cells are able to replicate indefinitely, and that's also a good way for us to study them into the lab, because we can take them out, put it into a petri dish, and they will just grow on forever. And unfortunately, they do the same in the body as well. And ultimately, as I mentioned, not all cancers, but some cancer cells tend to go into the bloodstream and invade other organs. And they have particular affinity for some organs versus others. And that's why certain cancer types have certain metastases in very specific loci and not random ones. But in reality, the field recognized at one point that cancer cells do not live in isolation. They are part of the host microenvironment. So you can imagine it's just not an isolated cells that decides what to do and that's it. In reality, this is more how it looks like. So you have some cancerous cells here that are next to the blood vessels, and we recognize their importance before. But there are also other type of cells that are the fibroblasts that compose the surrounding tissues, that are there are immune cells like T cells, B cells, macrophages that tend to fight against the tumors. There are fat cells, and we know that there is some association between obesity and cancer, for example, because fat cells can feed the tumor. So the field recognized this complexity and therefore developed updated hallmarks of cancer. So this is the same wheel as I showed you before, but now highlighted are the new ones that have been recognized back in 2011. So for example, the cancer cell and the host are fighting constantly. So the immune system tends to attack the cancer cell and get rid of the cancer cell, and the cancer cell responds to it continuously. And another point is that cancer cells can be supported by the host as well. So this process of fighting creates inflammation, and inflammation can be a driver and sustain tumorigenesis as well. Cancer cell needs to change their metabolism to sustain growth. So metabolism, that's been already mentioned before, it's been looked at a new hallmark of cancer, because cancer cell needs to accumulate nutrients, needs to ramp up their energetic demand and response in order to continue to proliferate. And cancer cells, as I mentioned, harbor mutations. So the starting point is oftentimes genetic mutation, not always, and I will go back to this point later. But really the combination of the different mutation is what sustains tumor growth. And mutation can happen at, let's say, 2 moments in life. We define germline mutations if they are inherited from the mother or the father. And these affect the entire body because they start as a single cell that divides. And as we mentioned before, changes in DNA sequence are maintained across all the daughter cells. So this implies that the whole organism will be impacted, and also half of the subsequent generation will be impacted. Somatic mutation instead happen at any point. This is very early. In reality, oftentimes they happen later in life, and they target only very few cells. One example that is readily understandable is skin cancer, right? If you're exposed to sun too much, you can get some mutation happening in your skin, and sometimes this will go on and develop melanoma, for example. And these will be limited to the skin, right? The rest of the body is fine. Whereas for germline mutation that I mentioned before, it affects the entire body, and we know there are some genes that are commonly targeted by mutation that are passed through generation. And I want to mention only some of them because they will be evident later on. So for example, P53 is a common one. Where is it? Notch1, I don't see it. It's somewhere here. Notch1 is another one. NF1, I will mention it later, is another, and so on and so forth. So these are genes that or BRCA1 and 2 that is associated with breast and ovarian cancer. These are genes that are often mutated and are passed through generation, and this increases their risk of cancer. This doesn't mean 100% necessarily that someone harboring mutations or variants in these genes will have cancer, and that's an important thing to note. So we know this is a census in the US population. We know that there are some commonly mutated genes, some genes that are most commonly mutated in cancer than others. So they are hotspots. And the top hit is P53. That is the most commonly mutated genes in human cancer in general, in the human population in general. And the reason for this is that these genes is tightly linked to the processes I mentioned before. So it's a gene that allows the cell to evade death and to continue to proliferate. So of course, cancer cells that target it have a growth advantage compared to cells that do not target it. So as I mentioned, genetic is one thing. All of these processes emerged as important. Another aspect that is emerging is that we are not alone. So whenever you're feeling lonely, remember that you're not alone. So all the tissues in the body harbor microbes or biofilms that in reality constitute the majority of our cells. 95% of the cells in our body are microbes in reality. They're not our own cells. So of course you can imagine that since they are found in every single tissue of the body, the most common ones are of course the colonic mucosa, so the intestine. We all know that we have a microbiota, but in reality they really can be found across basically all tissues. And you can think that they do something, right? We know that the microbiome is affecting how we digest, for example. And similarly, biofilms in other tissues affect how the tissue behaves, right? Mostly by altering metabolism. So again, it's another way, together with epigenetics, to respond to the environment. So everything that we internalize, I'm giving here the example of the intestine because it's more obvious. Everything that we internalize passes through the intestine, where there is a microbiota that modifies the metabolites that it comes into contact with and releases other molecules. And these molecules go in circulation and affect our entire body. But we mentioned a bit before, what is metabolism? Metabolism is simply a process, a set of chemical reaction that allows us to use some molecules derived from food, water, oxygen, carbon dioxide, everything that we come in contact with, process them, internalize them into the body, for example, to make up DNA, RNA, protein. Every single thing that constitutes our body is a molecule that derives from the external environment. And then, of course, it produces, it catabolizes, sorry, the word is missing, residues that it exports that are not needed. For example, carbon dioxide through respiration, water through perspiration, heat and energy, and urine and feces, of course. For everything that we don't internalize or that we process, we produce some residues that we export. And as I mentioned, microbes or biofilms have been associated to diseases every time you travel. I hope not every time, but it may happen that when you travel, you feel sick, right? And the reason for this is that you are exposed to things you're not normally exposed to, and your microbiome is responding to it. And microbes and biofilms are different across each and every one of us. So we were discussing over the break, you can imagine how this impacts our health. Like each and every one of us is unique, and also our microbiome is unique. So of course, all the processes that I described before and all the therapies that we may get, of course, will be modulated by the microbiome, so they will be different. And microbes or biofilm can change over a lifetime. So the microbiome I had when I was a baby are not the same that I have right now. And microbes or biofilm have been associated to disease. So one example is cancer, but we were discussing over break about brain disorders or also diabetes. They affect basically all of the disease because of how they process metabolites. And we know metabolites affect disease, so it's quite obvious. So for this reason, there is this new and special ad here since 2 to 3 years now, that is a focus on microbiomes and how they affect also carcinogenesis. So there are other things in here that I want to point out that emerged. That is unlocking this phenotypic plasticity, which is a complicated term, or non-mutational epigenetic reprogramming. So here we go back to epigenetic. And I want to stress how each and every single hallmark that has been identified and is identified over and over again gives us a new opportunity to target cancer, because we know that cancer cells rely on these processes to become oncogenic and to cancer to growth. So the more we know about these processes, the more we have ways to target. So the recognition of the immune system before opened up an entire new field in cancer therapy through immunotherapy, for example. The recognition of how the microbiome affects therapy allows us to understand why certain therapy worked for some people but not others. It can be that the microbiome affects how we process the chemistry of the therapy itself. So it's really important that we, over and over again, we understand how complexity of the cancer really works and how we can target it. And one key aspect is epigenetic. So as I mentioned before, old dogma in the cancer field, cancer cell needs to have a mutation or a variant in order to trigger the disease, right? Easy. No, of course not. So we are discovering over and over again that healthy tissues harbor mutations or variants that are commonly found in cancer, despite never giving rise to cancer. So this is an example in skin. It's 1 millimeter cube of skin, where each and every single circle you can think of as a cell of the skin, and every color is a mutation. You see there are many colors here, and this is a healthy skin. And the size of the circle is how frequent the mutation is. So you can see a lot of purple here, right? Purple is notch 1. Notch 1 is one of the genes I mentioned before that is more commonly associated with familial cancer. So how can it be that this skin is healthy? We said it's healthy, it will remain healthy. This patient, this donor will never go on and develop cancer. Same thing for P53, which is more reddish. They didn't do this in very nice friendly colors, but the red is P53. It's again, top most commonly mutated gene in all cancer in human. We can find it here, but this skin will never go on and develop cancer. A similar thing has been described last year in human breast, where they found chromosomal translocation, so pieces of chromosomes entering other chromosomes, that it's something that we commonly see in more invasive breast tumors, in otherwise totally healthy breast tissue. So how can this be, right? If we know that these mutations are found in tumor, why do this tissue are healthy? How can they be healthy? So of course, epigenetics is the answer. So very elegant studies through the more recent years notice that the epigenetic status of the cell before the genetic mutation really dictate if the mutation has an oncogenic effect or not. You can think about it as, we said before, the book with the highlights. If the highlights are in regions that are not mutated, where the mutation doesn't happen, then the mutation is not red. Then the cell doesn't care, basically, right? And doesn't go on and develop cancer. So this is one important thing that we are recognizing now, and this means that we can target the cell before, if we can understand the setting, the epigenetic setting. We said that epigenetics can be modified, right? So we can act on it much better, much faster than we can do with genetic mutations. And then, as I mentioned later on, really really cancer cells, once the cancer cell of origin is established and cancer is starting to emerge, we know that it really needs to rewire its epigenetics, so to turn on and off genes that are not normally turned on and off in order to sustain tumorigenesis. Again, you can think about it as a cell that is reading its book, and its book, it's telling it do not proliferate too much. So I don't know what's going on. So if you have neighbors that are pressing you, then stop growing. That's what a cell normally does. In the cancer cell, those words are not highlighted. So the cancer cell just continues to proliferate. So here I'm just showing you some of the most common epigenetic changes in cancer cell. I'm not expecting you to read all of this, just to understand that there are a lot. So I mentioned DNA methylation and histone modifications before. So acetylation and methylation, all of these change in cancer. There are some that are most commonly found in cancer, such as hypermethylation, so increased methylation at some genes and decreased at other genes. Normally it's hypermethylated at genes that prevent cancer. So it silences genes that prevent cancer so that the cancer can go on. And conversely, there is methylation at promoters that usually repress cancer, right? And so on and so forth. And now I will go a bit about what I'm doing. So we said that epigenetic in cells can precede genetics. So my question was, okay, is it possible that we are born with an epigenetic setting throughout our cells that make us more or less susceptible to cancer? That's the focus of my work. And that could explain some observation that we have in human twins. So I'm giving here an example. These two twins have the same genetic mutation. That's a familial cancer. So they inherited this variant in the gene called NF1 that I mentioned before, that is associated with neurofibromatosis type 1, which is an oncogenic form, a cancer. And as you can see here, these are monozygotic twins, so they have approximately the same genome, right? They both carry this very strong mutation, but only one of the two you can see it's affected. This poor twin had to go more than 30 surgical operation. He is blind at one eye and losing sight at the other, right? So he's really affected, unfortunately, by the disease. The other twin is overall healthy. How can this be? If genetics is everything and both have this strong driver, then how can this be that one is healthy and the other one is not? So what epidemiological data in humans show us is that oftentimes we do see epigenetic differences preceding the emergence of the disease. But of course, in humans, it's really hard, as I mentioned before, to go and mechanistically study what's cause and what's consequence. So what I did is I used, again, to simplify the search, I did this research studying this particular protein, which is called the TRIN28. And in particular, studying cases in which you have a lack of this protein. You don't have sufficient protein. So what happens is that this protein normally is attached to other proteins that silence gene expression, so compact genes. When you don't have enough, it opens genes that were not supposed to be open. And the first discovery that I did is that this is associated with the emergence of cancer. Here you can see some slides. These are all tissues that are affected by cancer. This is the lung, this is the normal tissue, and this is how the cancer looks like. This is a seminal vesicle, and this is the normal tissue, and this is a cancer tissue, and so on and so forth. This is a leukemia, for example, just to show you a liquid tumor. So first thing, no one described before that this particular mutation gave rise to cancer, so we discovered that. But what's more interesting, we discovered that if identical individuals have this mutation, they can give rise to two very distinct developmental paths, where one is overall healthy and the other one is developing cancer. So not having enough levels of this mutation can really set your epigenetic very early in development to be more prone or more resistant to cancer. And now we have a mechanistic model that we can use to understand what is changing, what are the players that are imposing these marks. And since it is epigenetics and not genetic, because the genetic is the same, right? It's just the epigenetic that is changing, then we can target it. We can target it with specific drugs once the cancer is emerged and reverted, but we can also target in early life by giving them supplements or diet in order to make a cancer-prone trajectory cancer-resistant. Or we can identify the more cancer-prone trajectories and design proper follow-up to inform medical decisions better. So I mentioned a little bit about possibility to target, right? Can we target cancer? So there are many of the classical ways to target cancer and other ways that emerged over the years. Oftentimes now what we use is a combination of therapies because we found that it's more effective. A classical one is the surgery to start off, followed by the radiation or chemotherapy. And of course, in case of liquid tumors, we have the opportunity of bone marrow transplantation as well. I mentioned a little bit about immunotherapy. So recognizing that the host and the cancer are constantly fighting give us the opportunity to act and modulate the immune system to make the cancer more visible to it and more targeted by it. And there is this section called here targeted therapy, where really epigenetic therapy lies in. So the idea is really to understand how molecularly the cancer looks like, not only in terms of genetics, because we understood it's not everything, but also from the epigenetic level, so that we can better design ways to target it. So with regards to epigenetic cancer therapy, we have many different opportunities that are now in the clinic or understudied, and they touch upon some of the systems that I mentioned before. So one of the most commonly studied and actually now in the clinic uses DNA methyltransferase inhibitors. These are the proteins that are involved in the methylation of the DNA, so compaction of the gene. And the idea is that, as I mentioned before, oftentimes cancers silence genes that are that stop it to grow, so that it can grow indefinitely. And this is just one of the example of what it does. So if we target the enzyme that continue to add this silencing mark on those genes, we can free these genes and we can tell cancer cells, okay, now it's time to stop growing, right? Like what a normal cell will do. Histone methylation is another one. So histone, again, are the proteins where DNA wraps around, and methylation usually is linked to repression. So it's another way that cancer has to silence those genes. And we have some molecules that are targeting specific enzymes that act by imposing this methylation on the histones. And acetylation is another one. Acetylation is the opposite, as I told you before. It tells genes to open up and be read. So if a cancer gene has a gene that tells it to grow indefinitely, it has it open so that it can continue to grow indefinitely. Now we want to close that particular gene. And now we have therapy targeting molecules that actually remove these acetylated residues. And what happens in this case is that histones are poised to say, okay, maybe we don't need to stay open anymore. And then we have the emergence of this methylation profile that actually closes the genes, that these genes that we don't want open in the cancer. So we have multiple options. And here is a bit too much detail, but we have some drugs here that are actually in the clinic right now. The target and the mechanism, as I mentioned, every time you see this DNMT targets DNA methylation directly, and instead EZH2, EZH2 is one of the top one that we have to target histone methylation instead, and HDAC, all of these target acetylation instead. So these are the tumors for which they are used. One interesting thing and unfortunate thing is that we actually have more drugs for liquid tumors than for solid tumors. Epigenetic therapies overall don't work super well for solid tumors. We're still trying to figure out why and how we can exploit epigenetic therapy for solid. There are some, but most of them are liquid tumors. And I want just to stress the advantage of epigenetic therapy over conventional therapy. So first thing is that they are more targeted. I mentioned they belong to the targeted therapy approaches. And the reason for it is that they really rely on molecular mechanisms that are specific for cancer cells. So the classical chemotherapy is the least specific way we have to target the tumor. And the reason for that is that it relies basically on the ability of the tumor to overgrowth relative to normal cells. But in practice, it targets all the cells. Now we have better ways to actually beam and target the cancer itself, but per se, chemotherapy is a very general and lowly specific way of targeting it. Radiotherapy is slightly better because of how it can be localized spatially compared to chemotherapy. As I mentioned, oftentimes these are used in combination. But epigenetic therapy is really highly specific because epigenetic mechanism of cancer cells are more specific. And so the therapy doesn't target normal cell usually, or less so. So there is increased selectivity for this reason. And as a consequence, we often have lower side effects. This doesn't mean that side effects don't exist, unfortunately, but we have reduced side effects. So, yeah. Oh, yeah, sorry.
Does that mean the parts you have in yellow highlighted there do not use chemotherapy or radio
That's just-
radiation?
Yeah, that's just the advantages of the epigenetic therapy over the other two. So it's a description you see here. I'm talking about target specificity.
Yeah, right.
Chemotherapy is low, radiotherapy is moderate, epigenetic therapy is higher.
And
after
what is the
specific.
therapy?
That therapy is targeting those mechanisms that I mentioned before. So DNA methylation, epigenetic modification at histones, all of these are considered to be targeted therapy
Uh-huh.
because they're more specific. So as I mentioned, chemotherapy relies only on differential proliferation of the cells.
Mhm,
Radiotherapy, it's similar, but we can target the beam better.
mhm.
So this is more specific, but epigenetic therapy is highly specific because the marks of the genes, where the genes. So I mentioned before that epigenetic in the cancer tells the cancer not to die, for example. And that's very specific of the cancer. So you don't find the same mark in normal cells because normal cells
Right.
die. And so they turn on that gene. So this means that if we use epigenetic therapy, that will target only cancer cell and not normal cells because it targets that specific mark. It's not so precise. So in reality, there's it has, we say that it has higher target specificity and it's not 100%, it's never 100%, but it works better because it targets that specific processes.
But does it eliminate using the other two
Not
before?
necessarily, not necessarily. It really, the choice of what needs to be done depends on the tumor type, on the tissue that is affected, on the stage of the tumor itself. And I'm not an MD, so I cannot comment on every single, therapy that is designed, and it depends on the patients as well. So we need to recognize also the physiology of the patients and the situation the patient is in order to design the therapy. So oftentimes what you see is a combination of the different therapy, because unfortunately what the tumor does oftentimes is adapt to the therapy. So that's,
We have another question.
yes.
This is all fantastic. A couple of questions I've had lingering in my brain, and the epigenetic stuff may be the answer to understanding this. A couple of things have always mystified me. So if you cut your skin and it heals, it's not just fibroblasts, but it's often regular skin cells that end up doing that and can modify the scar over time. So somehow those cells are being turned on to grow and turned off when they're done growing. So this is something that's ongoing, whether you let the cells multiply. I'm a retired eye surgeon, so we also are very concerned about the endothelium on the back of the cornea. Damage to the endothelium can result in all the things that the endothelium is supposed to do, but can't. You're never getting any more endothelial cells than what you're born with.
Mhm.
And they die off as you age gradually, but thankfully we have have extras. I'm not sure about Moses, but probably his corneas were okay, even the thousands of years later. But so one of the things is, can you reverse engineer this to turn on cells to divide and grow like a skin cell would, and stop when it's done growing as much as you want it to grow, like a corneal endothelial cell? It'd be great if you can get those to divide and cover a defective area, but then shut down when their population gets to a certain number.
Mhm.
The third thing is always is that I'm worried, and I'm wondering if epigenetic understanding would help with this, is cutting off a starfish limb.
Mhm.
A starfish limb can take that cut off limb and grow back from that stump a perfectly new limb, including blood vessels, nerve connections, the muscles, the cells, the little suction cups, all those things from that cut off end. And the ability to do that is, understanding that would allow all kinds of great things to happen as far as therapeutics. Is this all epigenetics as well, turning on and off the DNA of every single, you almost have to have a cell de-differentiate and then redifferentiate? How do it know, as the old joke goes?
Yeah, these are all great questions and great examples. So I want to go back to the example of the scarring, right, of the wound. And it's actually interesting because it's an active area of research in the cancer field, because one of the ideas that emerged in the field is that cancer can be viewed as a wound that never heals. right? So it activates similar processes, but as you mentioned, it doesn't stop once the wound is healed. So it continues to grow. And there are actually association in skin, for example, between wounds and cancer emergence, because the wound healing process goes awry and it doesn't stop. And that's where cancer emerges. So there are for sure similarities. And of course, the example that you made about regeneration. So cancer is basically, again, regeneration that doesn't stop, right? It relies on processes that are shared in the development and shared across cells. It just doesn't modulate them correctly, or as a normal cell would do. So of course, the more we study these processes also across organism, the more we can understand the mechanisms that don't work in cancer, and we can target them. I will make another example. So naked mole rats, I don't know if you ever saw them. They're a bit ugly in my opinion. There are those, like, they look like I don't know, moles, but they are completely without skin and they are blind. They live underground, like in Africa. Then they live for ages and they never develop cancer. How can this be, right? That's an interesting model. Like, how can they avoid developing cancer? Another example is elephants. Right? They're big. So there was one hypothesis in the field linked to cancer that the more cells you have, the more you're likely to develop cancer, but because of numbers, right, and of chances. But elephant like go in the opposite direction. They have many cells and they do not develop cancer, right? So there needs to be these fine-tuning mechanisms that are epigenetic linked, and we know they are linked to epigenetics because they are the same mechanism that go on during development. So the more we study them, the more we have chances to target cancer for sure. Yeah, thank you. Yes.
What are the mechanisms for targeting, like the top yellow one on the right here? If it's not chemotherapy and not radiotherapy, what is it?
Okay, they are molecules. So like here on the top left, as I mentioned, right? I gave the example of DNA methylation. So imagine you have a gene that tells the cancer cell stop growing. This gene in the cancer cell gets methylated, so it gets compacted, it's not red. So cancer cell just continues to grow because it has nothing that tells it to stop, right? That's the normal situation in the cancer cell. So what we are using are inhibitors of the enzyme that put this methylation, this silencing on those genes. And these are molecules, chemicals that actually target the enzyme. And oftentimes what they do is that they limit its binding to the DNA. So they detach the enzyme to the DNA, or they send it to degradation. There are different mechanisms. And basically the protein is not there anymore, or it cannot work properly anymore. So what happens is that the stop growing gene, now it's not methylated, it's not silenced anymore. It gets activated in the cancer cell that now reads stop growing, and then it stops to grow, basically. It's chemistry. It's all chemical. It's all molecules that bind different protein. Their specificity derives from the fact that they are based, I haven't been specific, I'm sorry. So they are based on the structure of this protein. So we can design specific molecules that bind to the different areas of the protein. It can either degrade them or stop them from working, stop them from binding the DNA, send them to degradation, and get rid of this protein, basically.
Would another example of that be the anti-VEGF stuff, the
Yes,
endothelial
yes,
growth factor?
Yes.
So it
That's-
just stops blood vessel growth and
Yes,
kills the
that's not
cancer.
epigenetics. That's a soluble factor, but basically it's the same. It's another inhibitor. Yeah.
Hi, just a quick question.
Yeah.
What mechanism do you use? Is it a pill? Is it an injection? Is it surgery? Is it a laser? How do you start the process?
So it's not a laser. Oftentimes these are drugs. So it depends on the type of drug, but it can be a pill or it can be, powdered form. It really depends on the drug, but you can imagine it as a pill. It's a pill. Say it once again, sorry.
It doesn't depend on the disease, part of the disease that you have or the cancer. It's the drug, how it's best administered.
Yes, it also depends on the disease, on the cancer type, in the sense that it can be targeted different. It's a bit like low dose aspirin versus high dose aspirin, right? The composition and uh, um, like added chemical molecules are different, and so the molecule itself gets internalized differently and sent differently to different parts of the body depending on the cancer type.
If I can go back again to the basic mechanisms, you're talking about an enzymes that acetylate or deacetylate histones.
Yeah.
But those enzymes are coded by the DNA.
Yes.
So to change the specificity or the activity of these enzymes, wouldn't that require a mutation in the DNA itself for the gene for that enzyme?
So that's a great question. So you can target a biological process in many ways. You can either target the sequence, the gene sequence. So if you go and modify the genetic sequence behind the codes for these enzymes, you will cause a stable mutation, but it's hard to do because how do you do it? Like either you do it in the zygote, and so you affect a whole cell in the body, and you don't want to do it because it's a bit, you can, but it's not easy to induce somatic mutations. You need.
Just that maybe that may be the mechanism whereby the enzymes are changed
Oh,
in
yeah.
activity.
Yeah. No, absolutely. Sorry, I was misunderstanding. I thought you meant therapy. So cancers, actually, many of the mutations that are found in cancer target epigenetic enzymes, so that these mechanisms can go awry and can cause this different epigenetic pattern in the cancer, so that cancer can sustain itself. Yeah, absolutely. Okay. So here I just wanted to share that Van Andel Institute is really on top of clinical trials and epigenetic therapy for cancer. So it's part of a team that is called Stand Up to Cancer. I don't know if you heard of it. Is a multi-institute team. For our institute is led by these two fantastic scientists. One is our chief scientific officer, Peter Jones, Dr. Peter Jones, and the other one is our co-appointed Professor Steve Balin. And it's a joint venture with other institutes. It's a nonprofit, of course, where we are trying to design new therapies based on epigenetics for cancer. And the idea is really to team up basic scientists with physicians and all the way through nurses and everyone really involved, multiple layers of patient treatment in order to design better therapies for them. And Stand Up to Cancer has some clinical trials that are currently ongoing. You can see them here up on top. And the idea is really to design new epigenetic therapies, define clinical trials and patient cohorts where we can properly test them after having done, of course, proper preclinical trial work. And we have some very promising completed clinical trials at the moment. So with this, I will finish this second part. I would just want to summarize a little bit. So cancer is a disease of excessive cell proliferation and invasion. As we said multiple times, there are these signals that tell cancer to never stop growing no matter what and invade other tissues. It's classified based on the cell of origin and the tissue that it impacts. It grows thanks to these acquired skills that we call hallmarks of cancer, that really a cancer cell needs to acquire in order to continue to proliferate. And the definition of these hallmarks has changed during time and will likely further change, and hopefully will further change, because it will mean that we understand more and more what cancer does to emerge, and so we have new possibilities of therapy. These includes, as I mentioned, proliferation, death avoidance, modification of the host, constant fighting with the host, such as with the immune system, changes in metabolism, and acquisition of new genetic modifications. And genetic mutation, as I mentioned, can be defined as somatic or germline derived, depending if they are inherited from the parents or they emerge at a certain point of life. And the microbiome plays an important role in modulating health. So we really need, I think, as a field to better understand how the entire body modulates cancer and modulates therapeutic response to cancer. Epigenetic is a key driver. It can be targeted by mutation, as someone said before, and can generate inter-individual variability even in case the mutation is the same. So even in people with the same epigenetic mutation, we can still appreciate epigenetic differences, and these can drive inter-individual variability in cancer susceptibility. So a better understanding of this layer of information can inform proper patient stratification, follow-up, and therapy. And so good news is that cancer can be targeted in multiple ways, including epigenetics. So now we have another bullet that we can use to fight against cancer. And with this, I will be happy to take questions. I just want to acknowledge this is our group. This is my PI, Andrew Pospisilek. I joined him at the Van Andel, and then he moved. He's a chair of the epigenetic department at the Van Andel at the moment. These are all my colleagues. The students have been so lucky to mentor all over the place, in Italy, in Germany, and here in the United States. And both at the Max Planck and in particular at the Van Andel, we have fantastic course that allow us to do groundbreaking work. They are very expert, super expert in their own field, so they help us because we cannot be expert in everything, right? I do have some collaborators at the Van Andel, personal collaborators, external collaborators. I was so lucky to receive some funding. And if you're into QR codes, you can scan this and look at my papers. And thank you all for being here. Thanks.
Thank you, Ilaria. Other questions? Looks like we have a couple. Here's Bill.
I almost hate to ask this question. How is current politics going to impact the funding of this research?
Do I need to answer? Okay, so the only thing I can share is that unfortunately some clinical trials have been stopped at the moment. So patients have been, sent back from the hospital because clinical trials have been stopped. That's, I think, the most impactful thing that we have in the cancer field at the moment.
I'm guessing that some of the research that has been stopped, some people have invested their entire career in that research.
Yes.
And now suddenly the plug has been pulled.
Yes.
Wow.
We hope, things will get better soon. Put it this way. And think we'll be back.
We all hope that.
I just want to say that we, as scientists, we like reforms. I think most of us, of course, there are some that are not happy with it, but we like, changes. We're aware that changes can bring good. So I think the system, as a foreigner, I can say that I always admired the NIH system for how it works. It requires a lot of work and a lot of steps to get funds. And that's good because we have multiple layers of check of where the money go. It can be improved. So I think all of us, the entire scientific community is open to changes if it can be improved. So, yeah.
Question here.
Yes.
I'll ask you to connect the dots between two of the points that you made. Do you currently have some of your work in a clinical study? Okay, that might be a good example to choose. So you showed a slide where epigenetics were highlighted in yellow.
Yes.
And then there are some cancers that will respond to that. Can you give an example, perhaps the study that you're conducting now, of how you determined which gene, which protein had to be targeted, how it was detected, chosen, and what you're trying to do with the therapy itself? Presumably it's an inhibitor or some other compound.
Yeah, thanks for the question. So I do not have, unfortunately, a clinical trial ongoing yet because these are all new results. But I'm to get in collaboration with Dr. Tim Trish at the Van Andel Institute, we are actively study a patient cohort of multiple myeloma. And I mentioned this, that I've identified the fact that people with this same genetic mutation can have different epigenetic paths, different epigenetic trajectories. And this means that I've identified what we call a signature, so a set of genes that is different in their epigenetic profile between a healthy individual and a cancer-prone individual, right? And that for us is our starting point to understand how these genes actually modulate the process. So once they are, I said that they are set in early development. So you can imagine as a fetus in uterus, you already have these genes that are marked in one way or the other epigenetically. So now how do they sustain cancer later on? That's an active area of my research, but also how we can use them as diagnostic tools, because now we know that they are different. So this means that we can take blood from the people and look at their methylation level, in this case it's methylation, at these genes, and determine if they are more prone or more resistant to cancer. And the advantage of the signature that I identified is that it's present across multiple tissue types and it affects multiple cancer types. So now we can look across multiple cancer. We chose multiple myeloma for different reasons. Among them is that I've identified this signature in the multiple myeloma model. And we have a very nice cohort of multiple myeloma patients that's been followed up for many years. And we have therapy data and response to therapy data from these people too. So we can design diagnostic tools, follow up. We can understand why therapy is working in some people and others not, all through the lens of epigenetics. So this is what I'm currently focused on.
And in this case, once you've determined that it is, the problem is due to methylation, how do you choose a particular enzyme or inhibitor to negate that effect?
Yeah, so the most direct one is this DNMT, DNA methyltransferases, because these are the enzymes that impose the methylation. But of course, in patient care, it's different what you do before the cancer onset and after. You cannot think about treating kids or people without cancer with epigenetic therapy because it would be, too aggressive. So the idea is that we can follow up them more carefully if they are in the cancer-prone trajectory. We can design proper follow-up. We can modify their diet, as I mentioned before. We can maybe modify their habits towards exercise, for example, if we think that it's relevant and linked to the cancer type in question, right? So that's before cancer onset. After cancer onset, we know that this signature is still present, so we can act through epigenetic therapy. And as I mentioned, DNA methyltransferase inhibitors will be the first hit since it's DNA methylation.
Thank you.
Thanks.
I have a comment from a Zoom
Oh.
participant. Says that this seems to be a perfect platform for AI to allow for shortening the time frame for therapeutic development.
Mhm.
Would you comment on that, please?
Yeah, thanks for the question. I'm not an AI expert, but there is a new PI who started at GVSU, and he used to be a grad student at the institute. He's collaborating tightly with Dr. Tim Trish. And they are working exactly on that, to use AI methods to speed up drug discovery, because unfortunately drug discovery is still too slow that we would hope for. But also they're using it to better identify cancer in tissue samples, because we know that sometimes cancer is so small that pathologists simply cannot see it, or it's confused, or an AI can help in that. There are multiple papers out now that tell us that AI can help. The issue that I see with AI, particularly in the biology field, is that it's really dependent on how you train the model. And we know that data in the biological field are biased oftentimes, even if I shouldn't use this word. But we have good data from specific sets of the population. We don't have good data from other sets of the population. And this means that from a diagnostic perspective or from a therapy perspective, we will be able to target better one population than the other based on AI tools. But AI can learn, and we are expanding our knowledge on other populations too. So, yeah.
Do you foresee down the road universal screening of people for these markers once you identify this driver and can look at that?
Yeah, that's a tricky question. So we were discussing about this over the break. So the issue of all of this is that ideally, what we would want as a biologist, what I would want is that we could deeply characterize each and every one of us in all these aspects like physiology, epigenetics, genetics, and so on. In reality, this is extremely expensive. So I don't think it's doable in the general population, in general term. Here, I would say that epigenetic can help, and combination with epigenetics and genetics can help. So if we can identify less expensive and also more sensitive, that's another thing, more sensitive screens that we can use in early development, similarly to what's done now for genetic screens, right? That tell us who we should properly follow up, who we should better characterize and so on. That will be a more, an easier way to go, a more sustainable way to go for everyone, including the patients.
Hi, one of the areas where epigenetics has cropped up is in the reparations movement, where Blacks have pointed to the long history of deprivation as causing epigenetic changes that might cause some of their health problems. Is that something that has the ability to prove? I mean, is that anything anybody's researching, or I know somebody must be researching, they wouldn't be talking about it.
Yeah.
But to my mind, that might be a huge policy area where you intervene in the health and try to protect them from whatever's going on, right?
Yes, yes, for sure. It's an active area of research. I'm not directly involved into it, but yeah, for sure. It's ongoing. It's ongoing. It's a very good point. Yeah.
I want to thank you very much for giving us so much time
Thank you.
today.
Thank
and
you
we've
so
learned a
much.
lot. Thank you.